I'd like to thank Mary and all the organizers for, I want to thank them for asking me to do this anyway. It was indeed a challenge, but I decided to accept the challenge. And as I wrote the abstract, which I found out later I had to do, I got through and I said,
I didn't realize it either.
I said, I've done so much. It's quite variable. And since I work on viruses, I can call it viral, but it's also in the other connotation of viral too, because everything I do seems to go viral. So, in terms of taking my time. But anyway, it's been a very fun career. I thought I'd go back a little bit to where I started. And like Jim and Charlie, I started on a farm in Indiana. My dad was inherited farm, family farm, about fourth generation. But his father died when he was 5 years old, so my grandmother raised him as a single mother and ended up in Chicago, where he went to high school, and then he went to Purdue in general ag. Then he went back to the farm. So he was a little bit more of a scientific farmer than the other ones in the area, which they kind of resented a little bit. But I remember as a kid having this smelly sulfur kind of barrel in the basement that he used on the orchard. And I wondered why. And other things. And one of the first jobs that my brother and I had to do was go down a long row of corn with a hoe in our hand. So I know what you mean when I'm doing that. And in fact, when they ask on the hill, why do we need Roundup Ready? I said, have you been out in the cornfield chopping weeds? I have. And I'll let you go do it if you want to. Anyway, I was the third of 5 children. My sister was the oldest. 2 brothers up until Pearl Harbor, and then my dad went in the service, and my younger brother Jim is our post-war model. So anyway, we had a bunch. We all had chores to do. We did a lot. Being the third, my sister and my brother got more of the field, and I got the yard and the garden and things like that. But we all worked together and had our chores. We went to a country school, and we hopped on the school bus and went to. The largest class that I had was 5. In the whole school, when they closed it down, there were 40 kids in 8 grades. Then I went to the next one. I went for a couple of years, and then my brother and my dad transferred us into the county seat school so we could get some college prep course, and both of us went to Purdue. And we were very active in 4-H. I did all the projects over there, including we had a purebred Angus herd, so we showed cattle county fairs, took them to the state fair, spent a week at the state fair in Indianapolis, had a ball doing things, and got to know a lot of people. So I had a scholarship in 4-H scholarship. We needed it because the farm wasn't that profitable, and it was only good in 2 colleges, home ec and ag. And girls just didn't go into ag. So I signed up, got accepted in the College of Home Economics, thinking I'd go into food chemistry or textiles or something like that related to that. Halfway through the first semester, I said, this isn't for me. I walked across the street and talked to who we called Uncle Dave, who was a fraternity brother of my father's. He happened to be the assistant dean for instruction in the College of Agriculture. And he said, well, we just started this new major we call agricultural science. It'll be a challenging course. You can go either plants or animals. And, take a few courses. So I had a zoology course. We dissected a cat all semester long in formaldehyde, and I said, that's not for me. I'd had enough on the farm, enough of the cattle and the pigs and the sheep. We didn't have sheep, but chickens and everything. I said, plants are much nicer to work with. They don't smell, you don't have to pick up after them and everything like that. So that's how I got into plants. I also had a very good beginning plant science course from Sam Postaway to Purdue. He was a master teacher. And my advisor was Ralph Shea, head of the plant pathology department. So let me get a little water here. Yeah. So they put me in a lot of challenging courses, including I went from home ec chemistry to engineering chemistry and chemistry majors the first semester, and into math too. I made it in everything except calculus, and I flunked that. They didn't make me take it over. I took a advanced statistics course. But anyway, Ralph was very good in terms of advising me. I had a biochemistry course under Joe Cooch, who was a master teacher. One of the experiments in that was purification of TMV. ammonium sulfate precipitation or something like that. I don't know. I remember the big dialysis tube, and that. And then I worked some in that because we needed the money, and I worked in entomology and counting pollen grains. So I had a lot of experiences at Purdue. And when I was, they said, what can I do after that? Well, you can go to graduate school. I might add too, in my graduating class at Purdue, there were 5 women in the whole College of Agriculture that were there. Most of my classes, I was the only girl there, particularly the calculus class. That was terrible. He didn't like girls at all. So I was used to dealing with a room full of men. So plant pathology was pretty good in that. My first APS meeting, which I'll talk about in a minute, there were 5 women there. I was the only woman graduate student there. So I made it through that, and Shay called me up one day and said, "We think you got a good background to go into virology." And I remember saying, "What's that?" Anyway, I did. They said they checked with a few other places like Wisconsin and Cornell and that, and they didn't have any assistantships. And they said, "There's a young man at Nebraska that's doing some really nice work." And Bancroft, John Bancroft was at Purdue then. So I got in my 53 Plymouth and headed for Lincoln, going into the Wild West. And they gave me a little desk there that I could sit in, and I had a desk on my own, greenhouse, working on plants. Marion Brackey was the advisor, wonderful man. Ellen Moorhead Ball, was the first person to apply serology to the identification of viruses, wrote manuals through the APS virology committee and worked on techniques. And of course, I had worked with her. I had to work with the bunny rabbits too. Marion was the developer of sucrose density gradient certification. Virus bands here, one in this, how do you, 2 in that. So we built the ISCO, the density gradient fractionator. Bill Allington, sons, started the company. He was department head. He had 2 sons, one had polio, and he was a genius, and he started this and designed that. Myron had the idea for this and said, how do you put something in the bottom and separate what's in there? So I worked on the prototype, and we had several different ones, and said, this didn't work and everything. So we'd send it back out there, and they'd come in, and they'd have me test it out and everything. Because I was working with a virus that had 2 bands. Whoops. And we didn't know it was 2 viruses or 1 virus or aggregation. That was the first time we knew that there were viruses that had divided genomes and had an RNA packaged in 2 different capsids. So that, and I did something just for the heck of it the other day. Actually, I was trying to find a picture of the ISCO, and I found this one. I still have my old Model D. When I closed down the lab, I saved that. I don't know what I'll do with it, but for now, it looks just like this one. And if you Google ISCO density gradient fractionator, all the papers come up that used it. On Google, I went through 50 pages looking at different ones, all sorts of fields. Then I skipped forward. I got to 100 and quit. From 1960s until 2017. And you can buy one on Google, on eBay too. They're for sale there if you happen to want one. Surprised me. I also learned electron microscopy. This is the old, I keep hitting the wrong one. The old RCA Model 2. Sit there and crank and look through there at virus particles. We got a new one while I was there. And my assistantship for my PhD was running the electron microscope. And we got a new one, and the technician came in and did it. I got the job of helping put it together and align it, because that was when Myron's wife had their 4th child. So he wasn't there that week. So I had to teach him how to do it. Another instrumental mentor was Mike Daly here. And Mike was a physiologist, but a really brilliant guy and very conversant and everything. It really made you think. My first meeting, they took me to that, was my first meeting with Biloxi, Mississippi. It's a little bit better than your first meeting, Jim. I was sold on APS after that, even though there were 5 women there, and I said the first graduate student, but it was fun. And to get there, Daly and Ellen Ball and 2 other graduate students and one of them's wife got in their '53 Ford and drove to New Orleans. So it was quite an experience. Ellen was good in terms of advising. I went there thinking I might get a master's in plant virology, go into medical work. She had come from medicine and worked in one of the first polio vaccine labs, knew about that and said, plants are a lot more fun. So I stayed with plants, continued on for the PhD then. But Nebraska was a great place to be. Then I went back to Purdue, mainly because when I got out there wasn't a job anyplace, except one somewhere in the South, and I wasn't too interested in that. At that time, John Bancroft, the virologist, was going on sabbatical to John Innes. And when I went back to Indiana to visit my family, I would stop into Purdue because they wanted to keep track of me and see how I was doing. So they said, we might hire his replacement. So they did, and I spent that year advising his 3 graduate students and teaching a class. And that time, we did everything in virology, including injecting rabbits and bleeding them and everything like that. So we learned the whole thing. And I can't remember here whether Bancroft was putting the swinging bucket rotor in to show me or whether I was showing him, because he hadn't done this much. And I said, I won't come there unless you order an ISCO, a fractionator. We had the first one in Nebraska. The second one went to Bob Fulton's lab at Wisconsin, and they sent me up there to teach them how to do it. I went to Puerto Rico to show Julio Bird how to do it, and it was at Purdue. It also was very good to separate polyribosomes, and we used to get beautiful bands on that. Joe Key's lab was next door, and boy, he was really excited about that. So I did a lot of things. Went to VPI, got in Potomac Division. You've probably already seen this one down here. You recognize you, Charlie? He hadn't changed much.
You needed to trim that beard a little bit there.
I think this was at a Potomac Division at some point. But then I went to what was then VPI, and someone came to me, a virologist somewhere, and says, what is a Polytechnic Institute anyway? So that's one of the reasons that, not I didn't make them, but others, that VPI changed its name to Virginia Polytechnic Institute and State University, and that's why we go by Virginia Tech, because it's too much. Anyway, Houston Couch had just gone there as department head, came from Penn State. He had visited Nebraska when I was a grad student, gotten to know me there, and talked and everything, and I guess I must have impressed him. I don't really remember that much. And he was going to Virginia Tech, and he knew he needed a virologist. So they found a new disease called peanut stunt virus and justified getting a position there. And Kurt Rohn has been a very close friend and mentor and was really helpful. Genetics of host-parasite interaction, and Kurt and I worked together on a lot of things. Kurt told me just the other day, and by the way, Kurt is 96 now, and I saw him, had lunch with him on Wednesday. Great guy. And he said, Houston told us he was going to hire you before you ever came here. I didn't know that. So I went and had a very easy interview, and they hired me for a whole $10,000 a year. And by the way, at Nebraska, I only got $2,100 a year, too. So you beat me on both of those, Jim. But you know, guys with families got more money than single women did. Yeah, we didn't need much money at all, so that was it. So from then on, which you get percentage incremental raises, I started at the bottom level. So anyway, it ended up pretty good. I did so much, I thought I'd just make a timetable here. Starting in 1960 at Purdue, and some of the, I said in my abstract, I felt like I was present at the flood. There is a book in molecular biology that says present at the flood, and I saw that when I was writing the abstract, and I said, that's where I was in virology, molecular virology, because everything started happening about here. First year I was at Nebraska, they had a speaker there that said, U codes for phenylalanine. And all the rest of them, the genetic code, went through, decided how they do it. The big mystery was when they had a sequence of the TMV RNA, and by then we knew the sequence of the amino acids in the coat protein by protein chemistry, and they didn't match up, and they couldn't figure out why. They didn't realize then that the coat protein was only this much of the whole genome. That was the level of where it was when I started out. I think it's amazing to see that where it has gone as far as we have. So my job when I got to Virginia was to identify viruses in peanut. I'd never seen peanut grown before. Tobacco. Then I get into corn. Corn is a Midwestern. I knew what corn was. My first job, paying job that is, was detasseling corn. Funk Brothers Seed Company had that. We spent days out there, so I knew about corn. Then we got into wheat, all these other crops here. I got into grapes and everything. And of course, teaching virology. So it kept me busy. I got into a lot of diagnostic part. Regional projects were very helpful. S70 was a corn virus project. And in that one, that's when I got to play poker with Bob Toller and others. And poker was good. I was telling some guys yesterday, I said, in one of the early women's, this was, really in the height of women's liberation. And I was always called, I was the women's lib, but I said, I'm just doing what I like to do. And if you let me do it, I'll keep doing it. I said, what women need to know to get along with men is three things. One is to play poker. And my mother taught me to play poker when we were little. In the military, my dad was in the Army. We knew all the ranks and everything like that. He came home to visit us, so I could teach military with the rest of them. And the other one was sports, and we were big sports fans in my family. So, I fit right in with the guys. Probably not too many women did at that time, but I think that was helpful for me. So all of this, and I also went to London. I told Charlie a while ago, I saw that announcement for the London meeting when I was in grad school. It was 68. I didn't graduate till 65. I said, I'm going. And one of my fellow students says, how can you say that? If you haven't finished school, you haven't got a job or anything? I said, I don't know, but I'm going. I did. I met a lot of people there, got hooked up with the International Working Group for Legume Viruses, got to know those, could been to a lot of meetings to make connection on that. O.W. Barnett started a foraged legume virus project in the southeastern state. Again, you get to know all the people in there. The other person that visited in Nebraska was John Fulkerson. And John was with the Cooperative State Research Service in D.C., and he pegged me right then. I didn't know it. And he followed me to Nebraska and back to Purdue, so he kept an eye on me. And I got a call, actually, from Gary Strobel saying, John wants you to come to Washington. John was having back trouble. I said, I don't want to get, I'm too busy down here in Virginia. Well, hop a plane, get up here, and we'll see. Well, they talked me into it. And I said, they wanted me a year on an IPA. I said, okay, I'll do it 6 months, but I got to be home to teach. They needed someone that knew what nucleic acid was, and they figured a virologist was the first one, because this was the area right after the Asilomar and the conference. And getting here to virus diagnostics, ELISA was just started in '78, and through the Foraged Legume Project, Richard Lister had the first ever workshop on ELISA at Purdue, which I was one of the people that were there. And then OW and people like Gaylord Mink and others talked with Chet Satula, and AgDia was formed. That took a lot of pressure off the virologists in the country. So instead of everyone bringing their samples to us and stopping what we're doing and doing diagnostics, we could help AgDia get started, and a lot of our antisera was donated to that. I made a lot of antisera in my lab. Still have it, still works, even though it's 30, 40 years old. Still works at all. So from the last half of '79 to early '90s, sort of at the time John passed away, I was full-time with USDA for 6 months, and then they kept a piece of me, 25% from then on. Of course, I was still doing my day job over here and had graduate students and everything, but they compensated pretty well to get help in my lab. I also had sort of an unlimited travel budget, so I got to go basically to any of the early plant molecular biology meetings that were held. Was there when we first transformed plants. Was there talking to Mayor Dale Chilton when she designed the TI plasma to get the transformation and everything. So that's what I mean. I was present at the flood. It's an exciting time. So in 94, I was named a fellow, and then with the job with CSRS, I also traveled around the country to regional projects and to review departments. So I got to meet graduate students in the other labs and started meeting a lot of women too. And of course, they identified me as a role model and everything. And that was when really women started coming in and getting more into the society. So it was in this time. I was elected counselor at large and then president and in the presidential series. And then afterwards was involved in various groups in Washington. COFARM was Coalition for Agriculture Research. Crops 99 was a coalition of the agronomic societies, cotton, corn, soybean, and wheat, to kind of bring those together to help funding. And it was working with Crops 99 that I first met Kelly Eversole, and kind of followed that and got her. She got to know the plant pathologists as well, so that's when all that started. And as Jim said, we started the National Plant Pathology Board. At this time, I knew I'd been counselor at large. I don't know. I was the only one here that wasn't a president, and Cliff had actually been working with USDA at that time, and we were trying to figure out how we could fill in the gap after folks had passed away. So those were all fun times. But I look back and wonder how I did it all, because we worked on this in the recombinant DNA, all of the regulatory through the coordinated framework, and the OECD workshops in Paris. And Jim and I had a lot of nice meals in Paris and did quite a bit of work over there, trying to teach international people what plant breeding was all about and how sort of good developmental practices for plants. And those documents are still used today, but nobody listens to them, unfortunately. So, back to my real job. The Soybean Mosaic is the soybeans in Virginia were increasing as a crop as they were. Soybean Mosaic was one of the most important ones. Basically, Bob Goodman actually started this system at Illinois when he was still there before he went to Calgene, but they found that they had various isolates from around the world. Actually, they got them from the germplasm collection that were at Illinois and Mississippi. They came in because they're seed transmitted, but they weren't all the same. And Jackie, you were there and remembered some of that. And I had been to a meeting where I learned about the bean pathosystem with the strains of the virus and everything. And our breeder said, well, if we just cross these, then we could get resistance to all the strains in one variety. Turned out the ones that Bob had were all alleles at the same locus, so we started looking for others, and there's 3 separate loci that are involved here. Since then, and this was a lot of cooperation, and we also developed a system where you could pound a leaf on nitrocellulose, and Amr actually did a lot of that kind of work, because this was a type of resistance here. This was totally resistant, and this slowed it down so much that it was an effective type of resistance. So anyway, this work has gone on now with the soybean genome. We know exactly where these genes are. We know exactly what the genes are, and they're quite different. And we just finished reviewing this, and the one at the RS34 is a totally different type of resistant gene. It's a dominant gene, but has no NBS LRR genes at all. So we've really done a lot in carrying that system along. It was something I could do to keep them straight on the virology while I did everything else. So, beginning back to the award ceremony, this is one picture I had to show too. Now Jim hasn't changed a bit. Right? And at the same time in '94, Kurt Rohn was named fellow, and also Guillermo Galvez, who was a fellow graduate from Columbia, a fellow grad student at Nebraska, whose thesis was purification of soybean mosaic. So it's really kind of come full circle on that. And just to show you that I get my hands dirty and everything, all of the way we got to this work is by the breeders making hundreds of crosses and planting out in short rows, getting down on the ground and doing airbrush inoculation, then going back in, assaying, and then proving that the virus that it had was soybean mosaic to get a 2-to-1 ratio. So we developed a lot of methods to make liters full of juice, as Kurt called it, to inoculate, and then we started testing all of these by octaloady, which works, and then by ELISA, which was a heck of a lot of work. So we developed basically ELISA on a membrane and gave a membrane to the breeder, say, you make the spot out there, keep track of the records, I'll do the development. And we could really made a good high throughput assay. But this was a fun project. This is Sagai Maroof, who's the molecular geneticist. These are some grad students that have gone on to do good work, and Moss Baldwin, who worked with me for years. I think this is the last one here. My career went so long I had to put it on 2 slides. From 96 to 2007, this was covered the genomics and the pathocism work. After 9/11, I got called in on a lot of different projects in threat assessment and Doug Luster's committee and everything like that. Got into NIPTers with Jackie. We got into the forensics worked on that, emerging pathogens, biosafety, and Vitiver and I have written several chapters together on the effect of plant pathogens on humans. And the fungi are the worst, but bacteria next, and they're actually starting to get some virus work. Did you know that in your human gut you've got just as much tabamovirus as you do norovirus? About 50%. Pepper mile bottle is all over the place. Don't know where it comes from. No one will bite the bullet and say it replicates in your gut, but they're there. So anyway, it's fun. And then the last thing I've had fun doing, was the awards and honors. I just got off of that last year, and you can't see it, but there's Allison there, and Bob is back there, and Barb. So they were pods last year, and Allison is this year. So I've known all of you. It's been a great thing to do, and I thank you, and I thank APS for all the fun I've had. I think I've only missed two meetings in my career, because it's like come home. So, thank you.
We have time for a few questions.
Well, I have to back up one here because I forgot to say something.
All right.
This long line across here. That after I finished with APS, Virginia Tech got the IPM CRISP project, and about this time, a student that came into entomology from Jamaica came in and said, I think we've got tobacco etch down there, but all we have to do is spray and control the aphids. And we started working on Scotch bonnet pepper and did a management project in Jamaica, and then I got pulled into managing the Caribbean site, and then that got into the whole Central America site. That got even bigger, and by the time we finished in this part, this is the year I retired, but you notice this line goes clear out here. That's because Bob and Naidu, who were involved in this, and Judy Brown and Mike Diome, and were the crew of us, and I was the manager. I was the puppeteer moving these people around, and we got up to 19 countries in 6 different regions. And Amr, who was my student, is now the assistant director of the project. He's from Lebanon and still working on it. The last phase, I said, I've had enough. But we did write a chapter to summarize all the work that we did on the IPM CRISP. With viruses, I think it's the first time that viruses have really gotten as much attention in an IPM project. But I didn't have time to talk about that, then I almost forgot it. Okay. Now you can ask questions.
Yeah. So your first APS meeting, there were 5 women. So about what year did you actually see, that, what is it, 35, 40% now? Or? Yeah, membership's 35% women, but among young women, it's like 50-50.
Yeah, it really is.
Was there a turning point at some time?
Well, they kind of had to get into the chain. I can't tell exactly, but I'll say this. I went to the first meeting there, and I can tell you who the other 4 are.
Yeah, who were they?
Ellen Ball was one of them because we went down together with Nebraska. Hilda McGrath, Marguerite Benedict, and Frances Latterall.
Oh, wow.
All Potomac Division. Yeah. Yeah. So I got to know all of them. The best part of the meeting, though, was hearing Carl Murray-Morsch give a talk on Kadang disease. That got me hooked. He is such a dynamic speaker. I've known Carl ever since then, and I saw him last at the IPVE meeting in Arusha on his 100th birthday.
Did he have a
And
suntan? Did he have a suntan?
Of course. He's quite a sun worshiper. He was 100 in the meeting in Arusha. When that was 2012, yeah, and he since has passed away. But I have a picture of him with me, and I also have one when he came to Blacksburg in the early '60s. So I have a lot of bridge type of pictures on that. But he was a great guy. But the second meeting, I think, was in Corvallis, Oregon, and I would walk through there, and everyone would say, "Hi, Sue. Hi, Sue. Hi, Sue." I stood out like that, and everyone remembered me.
Yeah, it was kind of an advantage.
So I had to have a way to learn people's names and faces. And after a couple of years, the guys would ask me, "What's his name? What's their name? What's their name?" So so I used that as an advantage because I did stand out. Yeah.
So you mentioned your athletic. I remember that very well because you and I were on an extended review with the Department of Wisconsin.
Yes.
And you insisted you took the whole team to bowling that.
We did? Okay.
I was so impressed. Saw you with your
Yeah.
your wristband.
I started the bowling in Nebraska because Stan Jensen, graduate student there, talked me into to joining the bowling team, and the department had a team, and I said, "Well, I've done a little bit, but I learned," and we bowled, and they gave you one night out a week.
Well, I was impressed. I still remember it.
The other thing at Wisconsin was that I had actually Bancroft and Shea and others had tried to get me to go to Wisconsin. At that Biloxi meeting, I met Glenn Pound, who was at Wisconsin, and he looked at me and says, "Yeah, I kind of remember your name. Your application was really good, but I just didn't want to encourage you because women are really pretty compromised in plant pathology." No joke. The review that we went back on so many years later that you remember, I was the chair of the review team. And they didn't have any women on the team, and you guys said during the discussion, "Don't you think we should recommend that they hire a woman?" I said, "Sure." I didn't have to bring it up, they did. So, what year was it? It would have been early 80s, probably.
I remember they also recommended they hire a hematologist.
Yeah.
Well, I will just add to that because I was a grad student in Wisconsin, and I went there in 1976.
Okay.
And that was the first time there was a kind of a slug, a large number of women graduate students. And I remember, I won't say who said this, one of the men's faculty, we were in seminar, you had to go, and he turned around like this and he said, "Oh my God, we're being overrun."
Yeah.
I think we all have a story.
Yes,
we do.
And the nematologist is a woman, and she's still there.
Still there, yeah. But I was talking to Sue Cohen last night, and she brought that up, and she was in school in '76 too at the same time. And she said that some of the women said to department heads and that, you got a lot of women coming along, you're going to have to get some jobs. And I had a conversation with Wiley Garrett when he was president. And I had been going around and seeing it. Every department I went to, I met with the grad students. About half of them were women, and the department said the women are our best students. And Wiley said, don't you think we ought to encourage more women into graduate school? I said, they're there. You need to get jobs for them. And it was shortly after that that we set up the Committee on Women in Plant Pathology. And Cleo Darcy was very helpful in that. And Sam Smith also was very helpful. And he helped send out some surveys to department heads to kind of get the feel of it. We had some interesting results on that. And some departments that said, no way. I don't remember which one. So I can't say exactly when it was, but it probably was in the mid to late 70s in that time thing. I was hired in '66. I was the only woman and the only single member of the plant pathology department until 1980, when we got a woman.
Were you the first woman who was ever hired in
in a tenure track?
Yeah.
I've heard people say that, but I'm not sure. I'm not aware of any other. But I think Helen Hart was on the faculty. I asked Kurt that the other day, and he was. But in the modern era, I think. And there was also I have a friend who had been to grad school in Arizona, not in plant pathology, but she knew some people there. And there was I think her name was Alice Boyle at Arizona. So there may be some more. I'm not making that claim at all. Ellen Ball was ARS, and there were a lot of people in ARS. The thing about Nebraska too that I didn't know, and I found in our history, was that Charles Bessie was one of the fathers of plant pathology, and he's from Nebraska. And Bessie Hall is a botany building, and there were the Walker sisters that were there. One was a plant anatomist, and they worked in botany and some plant pathology. And I looked at the charter members of APS. There were 10 women. 7 of them were Bureau of Plant Industry. Yeah, because you wrote some chapters in Jean Rosano's book. Yeah, and I looked back at that. And there was one that was in industry in horticulture in Pennsylvania, and the other 2 were in Nebraska. And I didn't know when they sent me to Nebraska, I went then to the only place that there was a woman on the faculty that I could work with. So I just happened to be in the right place at the right time and have good advisors. And I guess do enough to and work hard.
Just quickly to add to that, there's 3 Bessie Halls in the Big Ten these days. Iowa, Nebraska, and Michigan State.
Yeah.
The CE Bessie came from Michigan Agricultural College. I think it was class of 68, 1868.
Yeah. So anyway, used to be at the meetings, I'd go out with all the guys and have a good time. In fact, I was at one meeting and it was probably one of the regional projects, and I was getting ready to go out with the guys, and another famous professor that I won't name said, "Who's this woman? I'm going to tell your wife." And the other guy said, "Oh, that's no woman, that's just Sue." So I guess I just really fit in.
Oh my gosh, well, thank you so much.
It was fun.
Let's have our 3 other Pod Talk speakers stand up and give you guys all 4 of you a round.